Hey, it's about 10:30, so we'll get started on this presentation. Welcome, everyone. Thank you for being a part of Abiosciences and 10x webinar. We estimate this webinar to be about an hour and 15 minutes. Please type in the questions that you might have along the way. The panelists will answer it in the Q&A section. In the next slide, I will review the webinar's agenda. I will take about 5 minutes to present the overview of Abiosciences and what we do here. I will then hand it off to Jeff to cover the main topic, detecting RNA and protein simultaneously to produce spatial results, which will take about 35 to 40 minutes. Naseem will present on her topic, obtaining single-cell data from FFPE archived tissues, which will take about 15 to 20 minutes. And then the panelists will take questions from the audience. The services provided by Abiosciences are best described in these three pillars: a genomics lab, a bioinformatics department, and a software solution. We will go deeper into each of the services offered in the upcoming slides. As a 10xGenomics certified service provider, we have various certifications to help our clients obtain and analyze single-cell data. Abiosciences focuses and specializes in single-cell sequencing and single-cell data. Where other vendors might not work with low-quality samples, we provide help and assistance to our clients even with difficult samples. Our expertise lies in single-cell and spatial transcriptomics. Our bioinformatics team has tremendous experience in handling single-cell and multi-omics data. Abiosciences has helped many clients to publish in leading journals. We offer a cheap and effective way for our clients to utilize our publicly recognized and accepted pipelines for data analysis, which we offer in standard analysis and advanced analysis. In the advanced analysis, client can specifically choose from any of the following services based on their project needs. We also work closely with our client and regularly meet with them to update them on the progress of their project. Lastly, our software solution has the largest single-cell data set in the world. Users can find curated published data sets in their respective field. We can also add published research papers into our software based on the requests from our clients. You can contact us by going to our Contact Us page or scanning the QR code here. Or you can schedule a meeting with us by scanning the QR code. Or you can email us in the given email below. With that, I conclude my presentation and I will hand it off to Jeff to present on his topic. Thank you.
Thank you, Vic. Really appreciate it. I've been looking forward to this. Thank you for hosting us. And thank you everyone for attending. Really been looking forward to being able to present on our Visium technology. So my presentation will be in two parts. The first part is going to include some slides where I will introduce the Visium technology and talk a little bit about some of the new assays that we can do where we're looking at protein and RNA simultaneously. And then once I'm done with that, we'll switch over and I'll do a demo of our Loop browser software and show you what it looks like to kind of browse the data on both the RNA and the protein side, just to give you an idea of what those data look like and how they turn out. So, I'll put that all into my 35 to 40-minute section here, and then I'll turn it over to Nassim, and we can hear about how you guys do it. So I'll go ahead and start sharing my slides. So at 10x, this is kind of our driving mission statement in many ways, and it's assessing the complexity of biology. And we make tools to do this. And if you think about the complexity of biology, it's a little bit daunting. And it's pretty amazing that humans have been on this trail for so long in this complexity. I think it says a lot about our, optimism and curiosity and desire to understand more about the way the world works. If you think of something like the human body, for example, there's 40 trillion cells in the human body, and each of these cells has its own function, has its own set of expressed genes. Then within each of these cells, there are millions of interactions, millions of molecules, millions of molecular machines. These are all working together. They form tissues, which form organs. Each has its own unique population and diversity of cells. And so that means that at any one point in time, the organism can be in a variety of states. And our goal then is to design tools that can understand this complexity. And the good news is that we're in an era where computational biology, molecular biology, biochemistry have come together to make it possible to start doing this, to really interrogate biology and its complexity. And so that's what we're doing. And it's important to be able to characterize individual cell types and to understand what's happening in each one of those cells. And we've been doing this for a long time at 10xGenomics, where we've been doing single-cell RNA sequencing. But it's also important to put that in the context of the morphology and the spatial arrangement of those cells. And so we've recently been releasing platforms that allow us to interrogate biological complexity on the molecular level and then add to that the spatial context. And for a long time, these two worlds have been separate, imaging and molecular analysis. And, I come actually from the imaging world. My background is in doing a lot of image analysis and microscopy. And so to me, biology has always been spatial. And so this term that we use now, spatial biology, seems almost like an unnecessary name, right? But I think if we're coming from the world of molecular analysis, where it's possible to look at, the whole transcriptome, for example, that's primarily been, on the kind of separate from imaging. So I always like to say to my genomics colleagues, welcome to space, right? Here we are. And we're finally able to marry these two together. And by doing this, then we can gain really valuable insights, right? So some examples are shown here. This is by no means exhaustive, but we can look at how cell-to-cell interactions impact the microenvironment in something like a tumor. We can look in the immune system and understand how immunological conditions are created and maintained. And we can also use this to look at more complex interactions like those during development, or in the case of applying this towards something like regenerative medicine, looking at response to injury and gaining cues from developmental biology that we might be able to use to create curative and restorative medical treatments. And so all of these things then, depend on our understanding of not just the molecular events that are going on and the molecular state and the cellular state of tissues, but also how they're arranged. And so we have multiple platforms that we use for looking at biological complexity in a spatial context. And these include our Visium platform, which I'll be discussing today, and then our Xenium platform, which was recently released. And both of these are designed to be complementary. On the Visium side, this system is designed to capture whole transcriptome level information, as well as protein information, which I'll share with you a little bit later, and is one of the features of this discussion today. And it's sequencing based. So we're looking at the whole transcriptome across a whole piece of tissue. And then we are doing this at about the single cell scale with Visium. So we're capturing on the tens of microns level right now. And then we're going to be pushing this a little bit further with some new technology that will be released soon, where we'll have a bit better resolution. But either way, the transcripts are assigned to multi-micron areas. And so we're kind of looking at a regional global expression across the entire tissue. And it really works well for unbiased discovery. There's no need for a priori knowledge of the transcripts that you're looking for. You can just do de novo discovery in an unbiased fashion with the Visium approach. Xenium is designed to be a more focused system that is more of a hypothesis testing instead of a hypothesis generating system. This is an imaging-based system, and it's designed to detect transcripts directly in the tissue without the need for sequencing. And so we're getting down to subcellular resolution. We're imaging at less than 50 nanometers with this system, and we're assigning the transcripts directly to the cells that we're detecting them in. So both of these systems then give us a way to look at the biological complexity in the spatial sense from kind of two different directions. So let's talk about Visium. And over the past few years, Visium has been empowering a lot of impactful research. I share this to show that we have quite a few people that are adopting this technology, but also to show that this is something that's a known quantity, and it's quickly becoming something that people are asking for. And so, in addition to the insights that you're getting, this is also something that more and more reviewers are asking for, editors are looking for it. It's something that is becoming an indispensable part of looking at the transcriptome and proteome of a sample. And people have been making some really notable discoveries. Some of them are shown here, and also doing some really innovative stuff. We design our systems to be pretty open. And so we're glad if you want to hack it and come up with some unique ways of using the system. Some examples here include one of my favorites, which is spatial mapping of the total transcriptome by in situ polyadenylation. So this group at Cornell decided that they were going to use a yeast enzyme to polyadenylate all of the RNA and then map it in the tissue. So that includes more than just the protein coding RNA. And they found some really amazing stuff. So this kind of goes beyond what we typically would do with this assay, which is, quantify gene encoding transcripts, and really takes it into a new area where you can discover some really amazing things about biology. So it's a known quantity, and it's gone through some development over the years. We, especially within the last year, we've added a lot of capabilities. One of those things we've done is we've added this CytAssit device that allows us then to start with tissue sections on histology slides, and then we're able to transfer the transcriptome from these slides onto a Visium spatial gene expression slide in the CytAssit device. And this gives us an improved workflow. So it's a more streamlined workflow in terms of working with the traditional histopathology approach. But we've also seen improved gene sensitivity and specificity with this approach. And we have a broader tissue type compatibility and broader access. So with the Visium assay now, we can access FFPE samples, we can access archived samples, we can also do fresh frozen and fixed frozen samples. They're all compatible with the Visium assay using the CytAssit. And the way this assay works is we start with tissues and that have been sectioned onto a glass slide. And these may have been done, many weeks ago, many years ago. They could have been done fresh. And then we label with either immunofluorescence or H&E staining, and then collect an image of those tissues. And that image then becomes our first output from this assay. Then we incubate with our whole transcriptome detection probes, and then we transfer those from the tissue on the glass slide onto our Visium gene expression slide, the spatial gene expression slide in the CytAssit device. After that transfer is completed, then we extend the captured transcript probes and add the spatial barcode information, and then we sequence, and then we do data analysis and visualization. And so that becomes then the second output from the Visium assay. So you generate a tissue image, and then you also generate sequencing data. And then those data are pulled into our space ranger analysis pipeline, where we're matching the XY coordinates for the barcodes that are on each transcript to their location on the tissue. And then we output a loop browser file. And then in loop browser, we can explore the data where we have the expression information overlaid on top of our tissue image. And so we combine the morphological information with the sequencing information. And the data look like this. So here's an example using loop browser, where we're looking at, on the left here, global expression of the genes that were detected, the transcripts that were detected, and viewing that across the entire tissue. And then on the right-hand side, we're selecting regions of interest and then seeing which genes are being expressed here. This data can also be combined with something like pathology annotations. And so here we have an H&E section, an H&E stain section. This is a breast cancer section. And the ductal carcinoma in situ regions have been highlighted here in red. And what we can do then is we can select those regions and we can generate a spatial gene expression profile that matches that morphology. And then we can find that across the whole tissue. And we can make sure that we have canonical markers that we would expect that are being expressed here. But we can also go through and we can browse what other genes come out that are associated with this this pathology. We can also combine this with immunofluorescence. And so you can do IF with the Visium assay. And this allows you then to identify important compartments, important cell types based on protein markers from a visual standpoint. And then we can combine that. And this is in the same section. So this is not a serial section. And again, we can use those markers and we can generate clusters that are associated with different regions that have been identified using these fluorescent markers, or we can look at the expression there in those areas. And all of the data that comes out of a Visium assay is open source, so it can be easily run on a multitude of really nice tools that are available in the community. It's very easy to do all kinds of analysis, including multi-section integration and batch correction, single cell integration. So integrating with single cell RNA sequencing data, QC and filtering, normalization, variable feature identification, dimensionality reduction, clustering, deconvolution, where you are able to identify which cells are likely expressing what transcript in each spot. All of this data analysis can be done with some really nice tools. And so in addition to doing the RNA detection, recently, we have also added the capability to do co-detection of genes and protein expression. So using this CytAssit device, we have improved FFPE performance and transcript detection performance, but we can now also do co-detection of the protein and the gene information. So then this gives us 3 data types from a single section. So here we're looking at the morphology with the H&E stain, and then we can add on to that the RNA level where we're looking at the whole transcriptome. And then we can look at the protein level where here we're looking at an immune cell profiling panel. And we can then cluster these proteins that have been detected, and we can combine all of these data to give us a really comprehensive idea of what's happening in this tissue. And this is valuable because many times the RNA will give you a certain amount of information about what's actively happening, but there's a lot of functional information on the protein level that can only be obtained from the protein level because the half-life of proteins in some cases may be long, and the expression that generated that protein may be over, but the protein remains, for example, right? This is one good example of where looking at the protein level is really helpful in helping us identify the functional state of the cells in the tissue that we're studying. And so combining these data sets helps us to have that more comprehensive picture. The way this is done is that we have now on our spatial gene expression slides a capture sequence that will capture RNA, but we also have an additional capture sequence that captures antibodies that have been conjugated with a corresponding oligo-tag. And so our Visium spatial gene expression slides look like this, where we have these capture areas and we have these spots in those capture areas. Each one of these spots is made up of millions of these oligo capture sequences that are conjugated to the glass. And then when the tissue is brought into contact with this capture area here and this array of spots and then permeabilized, then those transcripts come out of the tissue and are captured. And now we have an antibody incubation step. And so the antibodies also will come out of the tissue and are captured on the slide. And then we add a unique molecular identifier and a spatial barcode to everything that's been captured. The UMI is unique per capture sequence. And so we know for the same gene and the same antibody how many of those were captured. But then this barcode corresponds to an XY coordinate on the slide. And so when we see a barcode, we can translate into an XY coordinate and we know where either the transcript or the antibody was captured on the slide. Right now, we've already optimized a human immune cell profiling panel. So this is a 35-plex panel. There's 31 immune cell focused markers and 4 isotype controls. And this includes cell surface proteins and intracellular proteins. This is for human and is very applicable in the immune and immuno-oncology space. And you'll see that it's comprehensive enough for this approach, but there may be proteins that you're interested in that aren't represented here. And if that's the case, we also have spike-in support. And so we have a couple of technical notes that are available that go through the process of acquiring and adding oligo-tagged antibodies for the protein targets that you're most interested in. And you can spike those into this protocol. And the way that that looks is something like this. So this is a slightly more detailed version of the general overview that I showed you before. And it gets a little bit twisty, kind of maze-like here, but I'll take you through. We start here, of course, with staining a tissue or sectioning a tissue onto a slide, and then staining, and then imaging. And then we proceed to hybridization with our detection probes. And then we do antibody staining. And so we're staining the tissue with these antibodies that have been tagged with these oligos. Then we permeabilize, and on permeabilization, we get both the RNA transcript tags, and we also get the antibody tags coming out and captured on the slide, and they're captured in place. And so we know that when we capture these, they come from that region of the tissue. And then we extend these, meaning we add the barcode and the UMI information. And then we go and we do our cDNA library generation, and we prepare 2 libraries. We have one library for the RNA, and then we have another library for the antibodies that are discovered. And then we sequence both of those, and then we do our data processing and visualization. And so with just this extra antibody staining step, then we now are capturing protein level information on a multiplex scale. And we can add here at this step, of course, our optimized panel. But like I said, you can spike in additional antibodies at this step as well. We can use something like the total-seq BN antibodies that are available from BioLegend. There are also some AbCam antibodies that are available. And then you can also conjugate antibodies. Both Abcam and BioLegend have conjugation kits that can be used for generating oligo-tagged antibodies that look something like this. And we're happy to give more information to those who are interested in this. But we have the antibody, and then it's tagged with this oligo tag that has these features here that are used for capture and identification. There's an antibody-specific barcode on each of these. We have a PCR handle for sequencing, and then we have this capture sequence that's complementary with the CytAssit gene expression slide oligo capture sequence. And so this can be conjugated onto the antibody of choice and then used at this antibody staining step. And like I said, we have a tech note for doing this. Okay, so now we can look at spatial biology in multiple dimensions, right? We're looking at the whole transcriptome analysis with high-plex protein and morphology from a single section. And using our platform, then you can run with confidence. We've already validated and pre-designed these panels. We have concordance with immunofluorescence. There's no impact to the gene expression results. We have novel automated isotype normalization that's in the that's already built into our pipeline, our analysis pipeline. It's easily integrated, and the folks at Abio have a lot of experience doing this sort of thing, and so they'll be able to generate some really high quality data. And I think that's where I'll stop in terms of the discussion of the Visium assay today, and then go ahead and transition over to doing a demonstration on our Loop browser software so you can get an idea of what the data look like. So if everything goes well, I'm going to be able to transition out of this. And then I'll share our Loop browser. Okay, what you're seeing now is the opening screen for our Loop browser. This is the latest version, 6.5. And the first thing you'll see here on the screen is when you open the software is you have a few sections. This section here gives you the version information, and you can click here and it'll give you an overview of what's new. There are some really nice resources that are built in terms of links here. So if you're new to the Loop browser, you can get started with the tutorial that's built in. You can go to the download page for data sets from Visium. I think the last time I looked at our web page, we have close to 80, I think, data sets from Visium data that you can download and explore just to get used to what this data looks like. We have a very nice technical support team that does a really great job of helping you use this analysis tool. And then, of course, we're always looking forward to hearing from you, so please use this feedback. And then over here, we have a list of files that have been opened recently. And when they were last opened, I was recently using this human breast cancer section. And so that's the one we're going to look at today. Once it's loaded, then the default is to show all of the clusters that have been identified, the gene expression clusters, and they're colored according to cluster, and then they're mapped onto the tissue here. And so this tissue was stained with an immunofluorescence stain. And so in addition to the RNA clusters here that are showing the transcript information, we also can see the fluorescence image underneath. And we can adjust this image. This has been stained with PCNA, which identifies highly proliferative cells. And then it's also been stained with Vimentin and DAPI for the nuclear stain. And while we're in Loop browser, we can adjust the fluorescence signal that we see here. For each of these channels, I think the DAPI is a little strong, so I'm going to bring that down. And then I'm going to bring up the PCNA a little bit. here. So we have a nice signal. And the pCNA is essentially identifying those highly proliferative cancer cells that are present here in the tissue. And we can do some things with this data that I think might be interesting. One of those is that we can compare the mRNA expression for that pCNA with the green fluorescence that we're seeing here. And so that's the first thing we're going to do. And to do that, then we can go over here and we can go and look for a feature. And so I'm going to search for pCNA. And we have two options. So it turns out that pCNA is included in our protein panel. And it's also included in our whole transcriptome analysis, of course, because if it's expressed, we're getting it. It's the whole transcriptome, right? And so if now we look at the expression of pCNA, then we can see, if we adjust the opacity here, so we can kind of bring in the expression of this transcript. You can see that the transcript does line up nicely with the pCNA expression, but there are places where it's missing, right? Where we can obviously see that the protein is here, but the transcription has stopped. And so this is kind of going back to my earlier point, which is that the mRNA can tell you a lot about the state of the system. And in this case, we can see which cells are actively transcribing and likely translating that pCNA. But cells that are already expressing and maybe don't have a lot of transcripts present, you're not going to pick those up from the mRNA side. And so you can see areas like these here where we don't see much of that. And so what we can do then is we can go and look at it from the protein side of things. So now I'll type back in pCNA and then go to the antibody. And now we can see here, we're going to change this to linear expression. It's a little easier to make out. And we can bring this up. And now you can see in these areas where it's especially dark on these spots, that is where the pCNA antibody was discovered. And we can do this and make it a little bit easier to see. So now we can see the pCNA antibody. And if we look then, if we kind of bring down the intensity here, that lines up very nicely with the pCNA fluorescence signal that we detected earlier, right? So now we're detecting on the protein level with this transcription-based assay, this pCNA. And so I think this does a pretty good job of showing how the two data types can work together to give us more insight into what's happening here. And of course, we can do this across all the features. We have tabulated information here for all of the transcripts that have been detected, as well as for all of the proteins that have been detected. So we can project those here with the Loop browser. And another thing that we can do with this version of Loop browser is that we can go ahead and we can look at, of course, all of the transcripts or all of the antibodies that were discovered, but we can also kind of segment. And so I've gone through and I've actually made some lists here where we can look for specific subsets of markers. So we can look at, say, cancer markers that have been identified across this tissue. So this is antibodies BCL2, PCNA, SDC1. And you can see they show very nicely in these areas where the cancer is at. We can also create a category T cell markers, again, using the antibody information. And now we can see where the T cells are infiltrating. We can look at where the B cells are. And we can look at the whole antibody list and where each one is being expressed. And so, Loop browser is a browser, right? And that's what it's designed to do. And you can see how it's very easy to browse the data and see where these proteins are being expressed. And we're getting, this is a 35-plex detection assay. So we're looking at dozens of proteins at once. And so it kind of takes us from this single color identification of the tumor with the pCNA to now having this really nuanced, multi-faceted, multiplexed view of the tissue. There are other things that can be done in here. We can recluster the data if we'd like. This is something that is available now in this newest version of the software. So if there are spots, for example, that may be less representative of the pathology that you're looking for, or maybe kind of fall outside the regions that you're interested in, then we can choose this recluster tool. And what it does is it opens a new window, which I think I may need to reshare on the new window. Let me do that. There we go. So it brings up a new window, and what you can do is you can actually recluster based on the barcodes that are present. You can use UMI thresholding, so you can choose specific numbers of UMIs, and you can set a threshold and kind of refilter using this. You can also threshold by features, and so you can identify different features that are important, and then you can recluster on that. And then once you have chosen the barcodes and the thresholds that you want, then you can recluster, and you can choose the way that you want to do the reclustering. And so this then allows you to have a bit more control over how the data are segregated, and then you can export this for further analysis. And so I'm getting towards the end of my time with you here today. So I think that's where I'll stop on going through this demo. But hopefully this gives you an idea of how the data look and then how they're acquired. You know, my goal has been really just to give a good idea of how the technology works and what the data look like. And, you know, please reach out to any of us here on this webinar, and we're happy to continue to dive into some of these details. So with that, I'll give it back for the next portion.
All right, thanks, Vic, thanks, Jeff, and thanks everyone for joining to our webinar. My name is Nasim, and today I'll be talking about the result of our research on obtaining single-cell data from FFPE tissues. As you know, single-cell transcriptomic has revolutionized the field of genomic, and it has numerous applications in different fields, such as cell differentiation, cell development, identifying novel and rare cell type, in disease research, such as cancer and cancer immunology. So in cancer immunology, single cell can be used to identify and characterize the immune population from different cancers. So for example, here we are showing to a study on human cancer. On the left, we see human kidney cancer, and we can see distinct populations of myeloid and lymphoid from tumor and blood samples. And on the right, we see immune population and subpopulation from human liver cancer. So this can lead to identification of new therapeutic target and new biomarkers for drug discovery. So these studies has been done on fresh frozen samples, and actually single-cell RNA was only applicable on fresh frozen samples, and many archived tissues like FFPE samples, which are fixed in formalin and embedded in paraffin, were not accessible for such a modern technology. But in 2022, 10xGenomics released a new reagent enabling obtaining whole transcriptome, fixed RNA from fixed samples like FFPE samples. And in this study, we try to obtain whole transcriptome from FFPE samples which are 10 years old, and use them by two different protocols to do nuclei isolation as well as cell dissociations, and later profile them to fixed RNA profiling kit from 10x to provide the gene expression library. So our study design, here you can see on the left the tissue block of FFPE from 2012 from a male person at age of 63, and diagnosis is adenocarcinoma or human gastric tumor. In order to understand better and evaluate the fixed RNA profiling kit on FFPE tissue, we also conducted a single nuclei of fresh frozen of human gastric tumor, so the tissue on the right, to see how would the data from FFPE sample would compare with the single nuclei that we obtained from fresh frozen sample. And we also provided a special Visium data of FFPE tissues, the same tissue. And later deconvolute that by the data that we got from fresh frozen, single nuclei from fresh frozen, and single cell and single nuclei that we obtained from FFPE samples. Here on the top, you can see the genomic workflow that starts from FFPE samples. So we use the FFPE samples by two different protocols for nuclei isolation and cell dissociation. And later, these purified nuclei and cells proceeded to 10x chromium fixed RNA profiling through probe hybridization. And then they were loaded to 10x chromium chip to provide the gems. And then the libraries were constructed and sequenced by Illumina NovaSeq. And the lower part shows the genomic workflow to provide the single nuclei from fresh frozen samples. So after sample preparation and nuclei isolation, they were proceeded to 10x chromium 5' single cell nuclei RNA kit. And the libraries were constructed for, and then they were sequenced. We also provided FFPE Visium special data from these archive FFPE samples through CytAssit technique. So we heard about the CytAssit techniques from Jeff's presentation. I'm not going into the details, but just to remind you, this is almost a recent technique enabling transferring FFPE tissue from a regular slide to Visium spatial slides. So after tissue transferring, there is a probe hybridization and ligation step, and there is permeabilization step to release the ligation product to be captured by a specially barcoded oligonucleotide within each capture area of the Visium slide. And then the library were constructed and were sequenced. Data analysis. So bioinformatic workflow for the data analysis is shown here. So first, we used Cell Ranger to align the data from fresh frozen single nuclei and FFPE single cell and single nuclei to human transcriptome by using Cell Ranger, and the Space Ranger was used for aligning the Visium spatial data. Then Surat R package was used to perform some standard analysis, such as PCA dimensionality reduction, clustering, and UMAP visualization. Later, the expressed genes from FFPE fresh frozen and single nuclei from fresh frozen and FFPE single nuclei and single cell were analyzed for gene ontology enrichment. And then to provide precise annotation for fresh frozen and FFPE data, we integrated these data separately by a public data set of human gastric tumor. And later, we used this integrated data set to deconvolute the Visium CytAssit data. Here you can see in more details of bioinformatic workflow on the Visium data. So after looking at the spot qualities, the spots were grouped together based on unsupervised machine learning method clustering. And later, to understand the spots more and investigate them more, we use the deconvolution method through deconvolution through RCTD method. So RCTD needs a single cell or single nuclei as a reference, which is already annotated and has the cell type labels. It learns the cell type profile from the single nuclei reference and uses that to predict the cell types within each Visium spot. So it can identify what cell types present within each Visium spot and what's the percentage of different cell type in each spot. So the result of data analysis from single nuclei, fresh frozen, and FFPE single cell and single nuclei shows that we got higher percentage of mitochondrial gene expression from FFPE samples, as well as higher percentage of cytoplasm and extracellular as gene ontology for FFPE samples in both single cell and single nuclei, versus for fresh, we get higher percentage of transcription factor expression. The PCA and UMAC projection shows that the cells and nuclei are actually separated based on the tissue type and tissue preparation type. For example, on the top, we see the single cell and single nuclei of the FFPE somehow grouped together. And this part, the red part, is the single nuclei of the fresh frozen tissue. So they are separated based on the tissue type preparation rather than single cell versus single nuclei. So gene enrichment analysis also shows that the single nuclei of the fresh frozen and single cell, single nuclei from FFPE also sensitive to expression of different genes, different kind of genes. And you can see we got different terminology for FFPE versus fresh frozen samples for biological process and also cellular component. So to provide annotation for our data, we integrated the single cell and single nuclei from FFPE sample with a public data set of human gastric tumor. This is studied by CytAssit in 2020. And this integration result shows that our FFPE cells and nuclei are very similar to the data set, and most of the cells could cluster with that public data set, and we got annotation for almost all the nuclei and cells in the FFPE. So FFPE sample is mostly composed of stromal cells like fibroblasts, endothelial, also epithelial cells, and macrophage. On the other hand, the fresh frozen single nuclei were not very similar to that public data set of human gastric tumor. And we can see a distinct population, distinct cluster here of our fresh frozen single nuclei that did not cluster with the reference, with the public data set. Later, we used the integrated FFPE to deconvolute the Visium cytosist slide. And we could actually retrieve 6 cell types by this integrated FFPE data set. So we got distinct region of epithelial cells on the top, distinct region of fibroblasts on the lower part. We got hotspot of macrophage, some endothelial cells, also TCD8 and parasite cells. We also use the integrated fresh frozen with the public data set to deconvolute the cytosist, and we could only capture 3 cell types, epithelial, CD8, and macrophage. So we can say that the convolution of the cytosist with the fresh frozen could capture fewer cell types, but we can say that actually more immune cell type were captured by fresh frozen deconvolution. The other cell type like fibroblasts, parasite were not detected by using fresh frozen deconvolution by fresh frozen tissue, because these kind of cell type were they exist in the fresh frozen at very low amount, And they could not be detected by the convolution method because RCTD needs more than 50 number of cells to be run successfully. To summarize, we demonstrated for archive FFPE samples, now it is possible to retrieve profile information on high single cell resolution. And we believe this technology can help further to characterize FFPE archive tissue, which already have pathologist annotation, also clinical data with, for biomarker identification. We further compared the single nuclei data obtained from paired fresh frozen tissue. And this comparison shows that we got higher percentage of mitochondria as well as higher percentage of cytoplasm and extracellular gene ontology in FFPE samples. On the other hand, fresh frozen sample had higher percentage of transcription factor expression. PCA and UMAP projection shows that the cells are separated by tissue type preparation, FFPE versus fresh frozen, rather than single cell versus single nuclei. By integrating the FFPE and fresh frozen single cell or single nuclei with a public data set, we showed unbiased detection of most cell type in FFPE samples. Based on integration result, the FFPE samples were mostly composed of stromal cells, macrophage, and epithelial. On the other hand, majority of the fresh frozen samples did not, single nuclei fresh frozen did not cluster with the public data set. We further applied the FFPE single cell data for the convolution of paired spatial transcriptomic data, and the result shows a spatial organization of different immune cells and stromal cells, and it showed a successful example of retrieving insight from archive FFPE tissues. So that concludes my presentation, and thank you for listening.
Thank you, Nasim. Great presentation.
Sure.
So we'll go to the Q&A section now. So Jeff and Nasim, if there's any questions from the questions that you feel pertaining that would be helpful for the audience, please go ahead.
Yeah, thank you. So one question that came in, I think, while I was presenting, is regarding the types of tissue that can be analyzed. So the question is, can this be done in CSF and or blood in CNS disease? So the Visium assay is designed to provide a spatial context to the molecular data that we're collecting from the cells. And so with things like, I think cerebrospinal fluid and blood, these are liquid tissues. And so they would be suitable for the single cell RNA sequencing that Nasim touched on a little bit, but may not have as much relevance in terms of looking at these in the spatial context with the Visium assay. You know, maybe if there's a little more to this, maybe if you're thinking about looking at it in the context of solid tissues, maybe in the context of maybe infiltration into spinal tissue, for example, if you're thinking about CSF, or looking at blood infiltration, right? Mainly like immune cells, then maybe, but typically blood and CSF and other liquid tissues are something you would analyze with the single cell RNA sequencing. Let's see, we had a few come in. Thank you for typing these in. The protein panel only available for human or also mouse? I only see human panel on the website. Yeah, so the panel that we have optimized is for human, but you can spike in mouse antibodies. So that's why you're seeing the BioLegend antibodies, and we have support for that. So we have a tech note that you can pick up that goes into how this spike can be achieved. It essentially, we provide guidance on how to optimize the antibodies and then when to add them in the protocol so that you can do your favorite mouse antibodies. So that's perfectly doable. And then there's another one, is doing Visium antibodies only possible with CytAssit Yes, yeah, the CytAssit generally produces better quality data, and so we've released this and any future updates for Visium on the CytAssit device. Thank you for asking. And then finally, there's a question about single cell segmentation, and then what are the number of transcripts per cell. So the Visium assay is right now capturing transcripts at a resolution of about 50 microns. So each one of those capture spots is 50 microns in diameter. And so depending on your cell size, then you may have multiple cells within that spot. And so what we do then is that if we are interested in understanding which cells are present in a spot, we can do something called deconvolution, where we look at the gene expression profile for each spot, and then we can identify which cells are expressing which genes based on that profile for a given cell type. And then we can deconvolve what's in that spot. And so we don't really look at this in terms of transcripts per cell so much as we look at it in terms of transcripts per spot. And depending on the tissue, that can range anywhere from a few thousand up to 8,000 or 9,000 transcripts, typically, in terms of transcripts per spot. So thanks for asking. Can we go to a cellular resolution using Visium? How difficult is it to get a protein panel for invertebrate models? So I think I already answered that about cellular resolution. Yes, you can, and it just requires that deconvolution or using large cells, right, after the fact. And then how difficult it would be to get a protein panel for invertebrate models. We don't provide that, but if you have protein that you might want to use for invertebrates, then you can conjugate oligo-tags to those protein antibodies, and then you can validate it using our guide.
Hey, Naseem, Jeff, thank you for answering those questions. And Naseem, so based on presenting the information that you have presented in previous events, the popular question that we get is, can you explain why the cells are separated by the preparation methods, not by single cell versus single nuclei methods? Could you answer that so that the audience is better educated?
Yes, so the data from FFPE and fresh frozen were very different in many aspects. And so, for example, I can say that fresh frozen is single nuclei is capturing mRNA from the nucleus. For FFPE, we got cytoplasm and extracellular because we had and single nuclei for FFPE samples. So that explains why the expression of the genes are different in two samples, and also the tissue preparation is very different. For example, FFPE is based on the probe base. Fresh frozen is based on capturing the poly A tail. So these two are very different fundamentally for the tissue preparation. So that's why I think the separation is based on the tissue preparation rather than single cell versus single nuclei.
Okay, great. Thank you so much. Is there any other questions? I don't see any. Yes, I think with that, we can conclude our webinar.